Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I am the marketing manager at Medicinal Genomics and proud member of the team that puts on the CannMed conference. All right, we do have a big update off the top for today. The official CannMed 24 speaker lineup is now live on CannMedEvents.com. This year, we have a great mix of familiar names like Dr. Ethan Russo, Dr. Dustin Sulak, Zamir Punja, and TJ Martin, to name a few. And we also have a number of new faces like Kimberly Gwynn from the University of Tennessee, Dr. Janester Wilson King from Victory Rejuvenation Center, Tim Lefevre from Nalu Bio, and more. Check out the full list at CannMedEvents.com and stay tuned to the CannMed Coffee Talk podcast as we will be talking to the CannMed 24 speakers leading up to the event this May. By the way, ticket packages are still available for CannMed 24. Go to CannMedEvents.com to learn all about the different packages that we have available and all the amenities that come with them. And if you want to learn more about what makes the CannMed Innovation and Investment Summit unlike any other cannabis conference available, check out the link in the show description. Our marketing team put together a great blog that highlights all the ways that CannMed is unique. Our guest today is one of those familiar CannMed names that I referenced earlier. Kevin McKernan is the Chief Scientific Officer and founder of Medicinal Genomics. Kevin has pioneered the genomics of cannabis and hemp to build a stronger scientific environment for the study of cannabis-based therapeutics and using blockchain technologies for tracking and verifying cannabis genetics. At CannMed 24, Kevin will present Understanding the Prevalence and Risks Associated with Endotoxins on Cannabis Flower, which will include novel research he did to measure the level of endotoxins in store-bought cannabis flower. Our conversation covers what inspired Kevin to look into endotoxin levels on cannabis, the microbes that produce endotoxins and their effect on humans, whether remediation techniques mask endotoxin risks, and more. Before we get to my conversation with Kevin, I want to thank this episode's sponsor, Agilent Technologies. Whether you manufacture or test cannabis, you face the challenge of delivering high-quality products, increasing sample volume, and meeting emergent regulations. Agilent offers best-in-class cannabis and hemp analysis solutions featuring robust instruments, software, services, and consumables. Their team of experts can develop, implement, and optimize methods to get you up and running quickly. Learn more at Agilent.com. All right, and without any further ado, please enjoy my conversation with Kevin McKernan. Good afternoon, Kevin. Thanks for joining us on the podcast.
I don't know. I don't know where you guys find such trash to come on this podcast, but here I am.
Oh, you're too modest. You're one of our best guests, and you are our first guest. So,
all right.
They can never take that one away from you. So I'm excited to talk with you today about something that's a little bit different. Because in the past, your CannMed presentations and when you've come on the podcast before, talked a lot about sequencing, whether it's cannabis or mushrooms or even viroids. But this year at CannMed 24, you're presenting on something a little bit different. We're talking about endotoxins, which isn't just a new topic for you. It seems like it's a new topic for the industry overall. But before we get into how or why the industry should start thinking about endotoxins, I think it's important to explain what they are and why you became interested in investigating them.
Okay, so I'll start with the why first and then lead to what, well, no, let's do what they are first so people know why we're anchoring into this. So endotoxins are molecules that are found on gram-negative bacteria on their cell wall. All right, so they embed inside this lipid bilayer, and they're known to be really potent anaphylaxis agents if they're found in an injectable. All right, so when I used to work at Agilent, we had all this demand to make endotoxin-free plasmid purification kits. So we had a whole program and a whole technology platform where we built these magnetic beads that stripped endotoxins out of plasmid preps because they're if you need pharmaceutical grade plasmids, this is a real FDA concern. You got to test them for endotoxins and get rid of the stuff. But I never really thought about it in terms of cannabis. So honestly, I'd forgotten about it for like a decade. And then the reason they came to our attention is that I think it was on LinkedIn, there was a little bit of a back and forth going on about decontamination and mycotoxins. And I think Tess had some really interesting information saying, hey, we're not looking at the right mycotoxins. Fusarium makes them, bacteria make endotoxins. And then another group in Massachusetts was asking a lot of questions about, what happened in the pre-roll facility in Massachusetts where someone died. This has like created a big storm here in Massachusetts. The CDC is swept in to start measuring things. OSHA's involved. There's been fines handed out. So for those who aren't familiar with this case, I won't add the patient's name because I noticed the CDC documentation didn't do that. So I'll stick to their guidance here. But it wasn't a patient, I'm sorry, it was an employee that worked in a pre-roll facility in Massachusetts, had an allergic reaction in the facility and like died. And I believe she was a cannabis user, so that everyone was really confused by that. Like, if this person was allergic to cannabis, she wouldn't be consuming it. So what the hell happened in the pre-roll facility that suddenly made her have an asthmatic reaction? Now there's some debate whether they had the proper masks and the proper HEPA filters and all that. And I don't know where all that landed. All I know is that by the time the CDC got in there to try to measure this retrospectively, the facility had already implemented some better HVAC to try to address this. So they responded to try to mitigate whatever risk they could, and no one really knew what it was. Like, something airborne must have gotten to her. So let's clean the air. So that had a lot of people asking us, like, what is this? Is there some microbe that makes a toxin? Is the microbial testing to blame? Pre-rolls are notoriously higher in microbial content for whatever reason. They're either lower quality flowers that don't have bag appeal, or maybe they're the lower branches and there's higher concentration. We don't know the answer for that, but they tend to have a higher microbial burden. Maybe grinding them up is something that liberates more of them. And there is this possibility that they're often decontaminated or irradiated. Now, we don't have good labeling here in Massachusetts to know if that was happening here or not. So that was on the table. And we had a lot of people just asking questions like, what do we do for, we're not seeing patients die, but if the employees that are handling this stuff are dying, then you have to assume there may be some artifacts that are hitting patients and they're just not getting maybe reported as being connected to their cannabis use. So that got me digging into the literature a little bit on this. And someone I know who's an endotoxin specialist was like, oh yeah, you should know about this. I mean, the tobacco fields had to deal with this all the time. The tobacco cigarettes have endotoxin, and in fact, they don't think it's the tobacco that's killing people. They think it's the endotoxin. I mean, some small branch of people believe this, that the endotoxin's what's causing a bronchial inflammation, and it's not really the nicotine that's doing this. This is the other contaminants in the tobacco that tend to come along for the ride, can create inflammation in the lungs and COPD and asthma and a host of other problems. So that put me down a rabbit hole of literature reading into this. And then I stumbled across some literature that implied 40% of the bacteria on cannabis is gram-negative. So it should be endotoxin loaded, which shocked me. I went over to our informatics guy, Steve, and said, is this true? Like, search our database for everything we've ever sequenced. The one nice thing we have here at MDC is there's 1,000 samples of cannabis that have been sequenced, mostly stems, but some roots, some flowers, and leaves, where we can measure, like, what's the microbiome content of these things? And is it true that 40% of these things are in fact gram-negative? And we combed through that for about a day, and we're like, yeah, the guy's right. There's actually a whole lot of Klebsiella. There's a whole lot of these other gram-negative bacteria that we're finding in the microbiome data. So that person's spot on. what do we do about this? So that's kind of where the project started. Is we said, all right, why don't we get ourselves an endotoxin assay kit? We can run that on like a qubit, which is one of these portable fluorometers. That's probably the cheapest thing you could ever purchase for a laboratory. It's about, I don't know, $7,000 or something to buy this qubit. And we use those for measuring DNA concentrations and RNA concentration. They're kind of a universal Swiss Army knife, really affordable. So we're like, all right, we can run it on that thing and assess if there's any endotoxin on pre-rolls or cannabis. And that kind of evolved into asking different questions of like, maybe we should look at cannabis that comes out of the dispensary system because we know it's tested to have low CFU. And let's look at some stuff that's in the hemp market that isn't tested. And usually, in our experience, the hemp market in Massachusetts has really high CFUs. Everything from total use and mold, total aerobic counts. It's grown outdoors, probably using a lot of organic farming. So we went around and got some samples from hemp facilities, and then purchased some from dispensaries that had been through testing. We don't have any knowledge of whether they're using any decontamination techniques, but one would naturally assume that when you decontaminate these things, you prevent the microbes from growing, but whatever's there has endotoxin. So the decontamination doesn't get rid of that. And so it's quite likely that it would be remaining. So the hemp samples we picked up were like screaming hot, 3 million CFUs per gram, like off the scale hot, 30 times above the limit. And then, of course, everything we pulled from the dispensary side was clean, like so very, very clean, like almost zero,
Right.
100, maybe 1,000 CFUs at most. And then we went about running quantitative PCR on those, plating those, and then also looking at live dead with quantitative PCR, because one thing we noticed with modern canna was that we have this live dead kit that gets rid of dead DNA. And when she was, I think it was Ginny, I'm forgetting
Ginny
her
Glaros,
name.
yep.
That's it, Glaris, yeah. So what she did a nice study that she showed at CannMed last year, where she was using live dead PCR with Grim Reefer to look at irradiated samples, and she noticed that in some cases the Grim Reefer didn't get rid of all the DNA. It lowered it, but it still in some cases was testing hot, whereas the plating was completely blank. And that got us looking into, okay, is our nuclease not working? Are there viable but not culturable cells there? Is there something about PCR that's just the irradiation can kill the organism but not destroy the DNA enough, so some of the dead DNA is still around and the DNases can't get to it. We kind of went down this rabbit hole trying to figure out why can't we get the live dead qPCR to perfectly match plating after irradiation. What she, I think, concluded from that study was that, okay, what we'll have to do is enrich after irradiation to know if there's anything there that's viable. That way, we can use live dead PCR and a little bit of enrichment and perhaps sort this problem out. But it certainly left Tess and a few other people in the field like worried that if we build a pipeline in cannabis where people can grow the dirtiest stuff possible and then just blast it at the end, just because it can counterfeit a plating system, is that really safe? Like, what if it's loaded with mycotoxins and endotoxins and all this other garbage, and you're just finding a way to evade the detection tool that they're using in the industry by doing this irradiation thing? And then, of course, there's concerns over it's not labeled and the patients don't know about it. So it got us looking in that area a little bit more. And I don't know. That's kind of how the project snowballed.
Yeah. No, and thanks for that. And you did write up a great white paper, kind of summarizing this whole study, which I'll put a link in the show description so people can read it. And so, I mean, you mentioned the results, and there's a great graph in here that kind of shows that of sort of the endotoxin levels and then the pre and post Grim Reefer information here. And I mean, but when I look at that, and tell me if I'm reading this correctly, that it looked like on every sample there was a significant level of endotoxins. Is that right?
Yeah, it was. And that was kind of the shocking thing that we found, was that there wasn't a sample that was free of it. Now, it varied, several orders of magnitude between the samples, but the lowest sample was several orders of magnitude over the DCOS limit, which we're going to have to assess in the field like what that limit should be. The DCOS limit, for people who don't know, is it's a European standard for the amount of endotoxin that can be in breathable air. So they have it set at 90 EUs per square meter.
Yeah.
We're dealing with a gram of cannabis, which is 1,000,000 times smaller than a square meter. So and we're having numbers that are orders of magnitude higher on the cannabis. So I don't know if they've set a standard for like what that should be for an inhalable product, but I would assume that the inhalable product should probably be close to the limit for what's tolerable for the employees in the air. And there are some studies listed in there from the CDC and from a couple other parties where they went in to look at the endotoxin levels just in the air at certain work environments in the cannabis field. The CDC had one study that showed that 40% of the employees were exposed to levels that were higher than in the trimming facility that were higher than the DCOS limits. Another study looked at this in hemp and a couple other like lung diseases that were showing up for workers that were handling a lot of hemp. So there have been a couple other data points looking at this. It's important to know that when the CDC went into this facility in Massachusetts, that's what they began looking for. They began looking at for endotoxin levels. So their radar is up on it, and that's what got us looking at it. Is we said, all right, if that's what they're coming in to investigate, we should probably get an understanding of how much of this is actually just around on the flowers, because we can't really find a whole lot of papers that speak to that. We can find some things on tobacco and a couple studies that were done in these environmental exposure studies. So yeah, the numbers are very high. I don't know if this. Obviously, if this were so toxic, there's a lot of cannabis use out there. People would be dropping like flies. We don't see that. I think what may have happened with this unfortunate case is that this person may have had a pre-existing condition of some sort, asthma, and this just was the hair that broke the camel's back. It maybe exacerbated the problem, and we have to pay maybe close attention to patient use in this case. So maybe the medical field needs to think a little bit more carefully about this. I mean, the other thing that came up that wasn't really mentioned in the paper but we've been pondering since then is, well, can we use gram-positive bacteria as biocontrol agents to like outcompete this stuff so it's not around? And that's probably a really good idea. Bacillus is something that's commonly used as a biocontrol agent, and Bacillus is gram-positive. So maybe you flood the plants with Bacillus, so you can knock out all the gram-negatives, and then we don't have this problem anymore. So it may lead to better cultivation techniques. The only challenge with Bacillus, our PCR tests will hit it on the TAC amplicon. It won't hit it on the E. coli or the sex stuff. But the TAC plates from 3M will light up with Bacillus. So a lot of people are penalized right now for using biocontrol agents that might mitigate this problem because the testing tools they're using will pick it up. And so it's really ironic because they have this testing platform that tries to grow everything on plates, and it's penalizing them for using the biocontrol agents that could get rid of the gram negatives that, in fact, the testing platform you can then evade by radiating. It's this big, circular nonsense that we've got high TAC counts, so we'll radiate it, and it doesn't get rid of the endotoxin. Now you can put it on plates that basically don't show any growth, when the real answer may have been to actually put Bacillus on there that would fail your 3M plate, and you wouldn't have the endotoxin problem. But yeah, we have a lot to learn, I think, in the microbial space in cannabis and what we do for this. But there are some citations in the paper talking about what these exposure levels are like, what they learned from the tobacco industry about this, and how they try and keep those levels down. The tobacco industry is obviously a little bit different in that every tobacco cigarette tends to have a filter on it, which we don't necessarily have on the cannabis stuff, and we don't know how much of that is scrubbing the endotoxin out of the smoke. But they do run tests on side stream and stuff through filters to see how much is coming through in tobacco, and they can in fact see it. I've not measured tobacco leaves themselves to see how much is on that. That's something we may do next week when we get some new kits in. But yeah, the amount on cannabis flowers is high. It does vary orders of magnitude, but it all seems to be above these DCOS limits. And the next question is, does pyrolytics kill this stuff, right? The problem we have in the research field is that endotoxin is one of these things that when it's around, you can't autoclave your way out of it. It tends to survive autoclaves, which is like high pressure and heat. And what we're using to vaporize cannabis is like low pressure and heat, but it's heat for like a brief period of time, and it doesn't torch the whole flower. It just torches the tip. So there's possibility that a lot of endotoxin can come out of smoke. I mean, that'd be a really interesting study if people can figure out how to capture endotoxin out of smoke streams. Some of the people studying the vape pens would be, I think, wise to do that. There's actually literature of endotoxins in and tobacco vape pens.
Oh yeah.
That the tobacco process of people getting, I don't know how that happens, because I don't think they're extracting nicotine from plants. Figured that was just a chemical synthesis they put in those things, but they have noted endotoxin inside of tobacco vape pens. In which case, I'm really curious to know how much of this stuff actually ends up in the cannabinoid oils, because it's a molecule that looks like it's going to be more lipophilic, and that it likes to be in a lipid bilayer. So will it end up in the extraction enriched, or will it end up in some other fraction of the distillation process? A lot more questions than answers we have right now.
Yeah, and that would be particularly concerning because, I mean, as I understand it, a lot of times if flower is failing for microbial, sort of the solution a lot of times is to extract it or turn it into distillate, and then
Right. I always assumed that was the simple thing to do. Yeah.
Yeah.
Absolutely.
In which case,
I think Ethan Russo. Yeah, Ethan Russo, and I think maybe it was Jeff Raber. A couple of people have done studies on some of the risks of extracting for safety. And one of the concerns was pesticides and mycotoxins could enrich differentially in the extraction process. Like the cannabinoids might go from 20% to 80%, but you can see the pesticides go from parts per millions to parts per thousands. Like they would enrich to a higher degree than the cannabinoids themselves. And so they raised concerns over differential capture of those things. So those are all good questions that we have now for what this endotoxin can do. So yeah, again, we've not seen people dropping like flies from endotoxins, but there is this fatality here in Mass, and it has got the federal government looking under the whole production process here. And if we are at the stage of maybe descheduling or going to Schedule 3, and there's going to be more federal overhang on the cannabis industry, we probably have to start paying attention to what they're doing and start looking at the source of these contaminants. I mean, I'm really intrigued by the biocontrol agents with bacillus, just because if you could flood the plants with gram positives, then there's less likely for there to be an issue there. And likewise, there's I think a valid point to many of the points you've seen Tess on LinkedIn raise. It's like, look, it's never a good manufacturing process to do things dirty and mop up at the end, right? You want to eventually build some level of sterility into your grow. I mean, it can't be perfectly sterile. Obviously, plants need microbes, but there is such a wide variation in the genetics that can that in terms of their CFU that you get. Like the work from Zamir was really interesting in that he showed the total yeast and mold ratios were a genetic characteristic of the plant. So he had plants that would always genetically would just have higher yeast and mold burdens than other lines, and it was something that went with the genetics of what they're growing. So that tells you something about we can find a way to breed cannabis that's low in these microbial burdens, so that we don't have to mop up a mess at the end and wonder if there's mycotoxins or endotoxins that are coming through that we can't measure. I mean, there are good ways to measure mycotoxins and now endotoxins. It's just one of those things that doesn't shine through in every state. I don't think every state has mandatory mycotoxin testing, and no one has endotoxin testing. And frankly, looking at the data we have now, I'd be like, forget the mycotoxin, look at the endotoxin, because it's everywhere. When I talk to people who run the mycotoxin test, they're like, we almost never get a hit on those things. It's just like
and so testing. Why is it that mycotoxins became, something that's required testing, and endotoxins kind of went unnoticed?
It's a good question. I mean, mycotoxins made their way into the food industry, and they are more of an oral ingestion risk because they tend to have effects on your liver. Whereas endotoxin isn't something that's really an oral ingestion risk. So, like the edible pipeline doesn't need to worry about this. It's really more of inhalation and injection. In fact, injection is the real thing that the FDA worries about, because you get anaphylaxis and people drop. But inhalation is something I think has been one of these areas where I mean, I'll have to look up the pharma regs for like the limits on this for like asthma inhalers and stuff. I'm sure they're very low, but I think when you get into the realm of like tobacco and cannabis, you're like, okay, you guys are inhaling like, pyrolytics anyway, so who cares about about what else you're adding? I just don't think that they've had the same like scrutiny. It's almost as if you've committed yourselves to performing something risky, so you're probably not too concerned about the other risks that are there. And
Right.
there's always this question: the flame probably destroys it all, right? It's probably not true given the temperatures that people vape at, but it certainly adds an element of the pyrolytic seems to sometimes offer an out on what's present. So I have a feeling it has something to do with that. Just that if you're inhaling these, if you're willing to light something on fire, inhale it anyway, then no one's got a whole lot of sympathy if there's some contaminants around. But I think the medicine, when it comes to actual ingestion, it probably does we need to think about mycotoxins if they're at high levels. I've not seen a lot of people who test for those things, so they come out, more than like 1% positivity in testing. I always hear of like 1 in 1,000 or 1 in 500 samples might pop positive for a mycotoxin test, but it's nowhere near it doesn't match the level of like 10% failing for total yeast and mold. You'd think if you had that much yeast and mold everywhere, you'd have some concordance with the actual mycotoxin test. So what we can see from this data is that, yeah, when there's bacteria there, and even when it's been zapped or it doesn't show, like we can see in this paper, there is, in some cases, there is bacterial DNA present and it doesn't grow. That tells us, okay, someone probably decontaminated this, and the endotoxin is screaming high. So the DNA can maybe help navigate some of this. But when it comes down to some samples that are just totally, no DNA, no evidence of growth, but they're screaming high with endotoxin. That's, I don't know, that's another matter. And we probably have to understand what the lower limit can be from a patient standpoint.
Yeah, and the other point too, when you talk about sort of the pyrolytics or lighting it on fire might take care of the endotoxins, might eliminate them, but it doesn't solve the problem of the people who are working in the production facility or the person in
Right.
Massachusetts.
Yeah, and I think you had a podcast on with an aspergillus expert who always brings up this point that when they do these pyrolytic tests, that there's a very high temperature at the end of the joint, but the rest of the joint is getting an elevated temperature, but not hot enough to destroy the spores. So you can get active spores coming out of a joint. In fact, we've seen this in the Bakersville case where they had cryptococcus infection of a patient, and they ended up with meningitis. So yeah, the pre-rolls seem to be the part of the pipeline that's dirtiest, and where we see the most fails and where we probably need to have the most attention. I mean, that happened in Oregon too. When we looked at the whole Oregon story, when they turned on aspergillus testing, they were having like 10 to 20% failures in the pre-rolls, but their flowers were down at like 2 or 3% like California. And it's just a signal that we tried to make clear to the regulators that, look, you have a biological signal here that would satisfy a Bradford Hill criteria for causation. You have what's known to be a dirtier process for making pre-rolls from all the scrap material that's testing hot, and you have high quality bag of peel flour that's testing low. This is the sign of something that's going on. We should look into this, and maybe the pre-rolls have to go through a different decontamination process and maybe extra mycotoxin checks or something. But I think they've opted to just throw their hands in the air and move on. But I mean, the data that we're seeing here in Mass is it's not something we can turn away from. I think the main argument they had in Oregon is, well, no one's ever found someone who got hurt from this, and that's a fine argument if you put your head in the sand and you don't look. But we actually have a fatality here in Massachusetts, and no one was necessarily looking for it. The person literally had to drop dead on the floor at work for people to pay attention to it. We don't know how many people are getting asthma attacks outside of the workplace and just consuming dirty cannabis. It's probably going unregistered, and that is a problem with a lot of the cannabis literature out there. It's that for all the time of prohibition, we didn't have people running into the hospital declaring, "I'm a cannabis user, help me." They were hiding it because it could get their kids taken away from them. So the best we have right now are studies from like the CDC that point to insurance data showing that, okay, cannabis users have an elevated risk of fungal infections, and the ICD-10 codes like point to mucormycosis and aspergillosis as like the two top risk factors that are there. But other than that, it's a lot of people sort of claiming, well, we never had to do this during Prohibition, so why do we have to do it now? And that's fine if you want to socialize the risks of the business, but it's not necessarily, I think, where things are going to end up when the FDA gets involved, if they ever get involved.
Yeah. And so is that where you're kind of hoping is one of the results of looking into this, is that we should kind of start looking, or at least, consider that endotoxins might be a factor here?
I definitely think others should look into this. No one should take our work alone. It's a preprint right now. It
Sure.
needs to be reproduced, and it should be reproduced in multiple jurisdictions before any regulator gets crazy with this, because we don't know what this means. I think what we want to see is lots of reproduction in the academic space or in just in the scientific literature, and then get some assessment as to is this associated with other people having asthma attacks. It is of all the things that we can do in the cannabis field, this is probably the cheapest thing one can measure. And I'm sad to say we don't have any like revenue stream attached to this thing, because it is it's not our technology. We're just buying this stuff from Thermo. But it's very cheap. It's very, small format. I don't know if you guys can see this, but right there.
Your background's
weird.
fighting against us here.
Yeah, it is. I have to put it on my face for you to see it, right over my forehead, maybe. People listening can't see this, but this is basically a standard curve of a LAL assay. It's one of these assays that has to use an enzyme.
If you send me a picture, I can superimpose it up there for you.
Oh yeah, it's probably one in the paper. But yeah, it's so there's this clotting factor in the blood of the horseshoe crab, which is blue blood, that actually, when endotoxin shows up, it creates a clotting cascade. And so they've hijacked that enzyme to create this sort of fluorometric assay that basically gives you a different color based on the amount of endotoxin that's there. So this is something that runs in a portable fluorometer it's smaller than a PCR machine. It's probably the cheapest thing anyone could possibly put into a cannabis testing laboratory. And it's probably only $10 a sample or something. So it is really fast and cheap to run this. It's something that is run quite frequently in the pharmaceutical industry to make sure there's an endotoxin in any of the drugs and any injectables. So there's a large industry behind how to do this and do it properly. The one challenge that I think we've had with the assay is that you do have to run this in matrix, because the LAL assay, there isn't a really good way to purify endotoxin away from all the other stuff that it's with, which means you're having to take cannabis flower, homogenize it, dilute it. Really, we basically dilute it 1 to 10. After we've put a gram of cannabis in 10 mLs, we take 1 microliter of that and dilute it 1 to 10 and pop it into this assay, and we get these screaming high signals. But if you do it any more concentrated than that, the terpenes, or I don't know what, but something seems to be interfering with the assay, and you get an under recording of the event when you do that. But other than that, we've been able to go through probably 24 samples now of different flowers to show that we can pick it up with this portable device that is fairly cheap. The reagents are very time sensitive, so the moment you crack them, you got to use them. Otherwise, the assay starts taking off on kind of background endotoxin. So that's the only downside of the one that we're using, is you can't crack the vials and leave them out on the bench for 2 days and expect to get good results the next day. You have to kind of be really fast with it. But so yeah, I don't know where it will go. I hope the other side of this is that could be really interesting as a collaboration with a testing laboratory, is that there are other techniques out there that measure endotoxin with mass spec, and they're far more accurate and better. But they're far more expensive, and they're not portable. And but they're really good tools to validate, all right, is this really working the way we need it to with the cannabis background that's there? That's always good to have a second assay to kind of check the books on this stuff. And so to the extent any other cannabis lab out there has done endotoxin testing, please reach out. We'd love to share notes and how we could try to calibrate these things to each other, and see if there's there might be a way people can just turn their mass specs onto a different column or a different assay and read this with the equipment they already have. But it would be a helpful study because I think as more and more of these decontamination tools come to market, we have to understand what it is we're hiding.
Mm.
It's one thing to get rid of total aerobic count. Total aerobic count is one of these weird things where it's like, you don't really know if it's harmful or not. You're just counting total microbes. But, and when you start doing these cleanup techniques, you're getting rid of what can grow, but are you getting rid of what you're most worried about, which is the endotoxin? It doesn't seem like this is, we're blinding ourselves perhaps and giving ourselves a false sense of security with this. So this might be a tool that can help either guide that process. One thing that we're working on next on this, and this is actually, I have to compliment Tess. I'm forgetting her. I'm going to butcher her last name, but I think you know her from some of the great podcasts she's done and put on LinkedIn. She looked at her paper and was like, this is cool. You could do more replicates, which is always good advice. And then what about getting rid of the gram-positive bacteria out there? Would you get a better PCR correlation? If you look in our paper, our correlation between like qPCR and endotoxin is like 0.43. It's there, but it's not like you could use the qPCR to predict this. I think some of that lack of correlation is that we're not segregating the gram-negative from the gram-positive in our qPCR test. We're just measuring total aerobic count. So we've gone and designed some PCR primers that are going to try to do a better job of like only give me amplification from gram-positives and only give me amplification from gram-negatives. And then we can try to correlate the endotoxin levels with those PCR signals. And maybe those signals will like get a 0.6 or 0.7% correlation. And so the quantitative PCR test would not only count all TAC, but would split them into two different channels of positives and negatives. In which case they may have a little bit more predictive power to run after decontamination to know, okay, how much residual DNA is here that was gram-negative, because the gram-negative DNA is what we want to make sure is really low. That would tell us that the plant started with very little gram-negative, and therefore it's unlikely to have endotoxin. So we're poking around at some assays like that to see if they better inform on the topic. But to be honest, the PCR might be helpful in the end. The endotoxin stuff runs just as quickly. It's like a 30-minute assay, and it runs on a device that's much like smaller footprint. It's just hard to parallelize. Like we can't run 96 at a time without the reagents kind of going bad on you in the process of setting up 96 assays. So it's great for running about 8 at a time, and then you have to probably start over with another strip tube of 8. But the PCR stuff we can really crank through, and it might help us inform on all right, what's going on with the whole decontamination process.
Excellent. Well, it seems like definitely an interesting line of inquiry that we're going to continue to look into. In the meantime, is there other resources you would like to point the audience to that they can learn more about this, or if you want to share any ways that they can get in touch with you, please do.
Well, certainly, yeah. You can find me at this Medicinal Genomics at that address, and then I think I put a Substack up on this, but there's the preprint we have. I'd start there. I think the preprint has a good list of like, what have other regulators looked at in the cannabis field regarding endotoxin? What's in the tobacco field? What are the limits people have had in the past? And our sort of first go at this. And I would caution folks, we just started doing this in like November. So we're by no means experts at this, but we figured when the data came out, like as loud as it was, we're like, all right, we got to at least get this out there and let others know and see if anyone else can like tell us where we're wrong or which direction we should go. So if others are interested in studying this, like bring us up. We're open for collaboration on this because this is something that is kind of new to us, at least in the cannabis field. And we're kind of interested in where this may lead. So feel free to drop us a note at, you can get to my email at kevin.mckernan@medicinalgenomics.com.
Excellent. All right, Kevin, thanks again for the time, and see you around the office.
All right. Yeah. Take care.
I hope you enjoyed my conversation with Kevin McKernan. Check out the links in the show description to learn more about the topics we discussed. And thanks again to this episode's sponsor, Agilent Technologies. Our next episode drops February 7th. In the meantime, go to CannMedEvents.com and check out the CannMed 24 speaker lineup. While you're there, be sure to sign up for email alerts to get notified about future event announcements. And don't forget to register for your CannMed 24 ticket package. Tickets are limited this year and demand is high, so don't wait. Also, we'd appreciate it if you follow us on social media. We are on LinkedIn, Instagram, Facebook, and X. Just search for CannMed Events. And finally, we'd appreciate it if you rate, review, and subscribe on whichever platform you use to consume the podcast. All right, and that's it from us. Until next time, stay safe, stay healthy, and be sure to join us for the next CannMed Coffee Talk.